here ever had a genetics course? A couple people kind of, yeah. We don't require it. Moving forward, it's one of those classes that we might bring in because so much of our world is going genetic in the laboratory. We do have an introduction to molecular diagnostics that you don't take until your third semester. We talked about maybe moving that to second, but that's still kind of in limbo. So we don't get a lot of genetic stuff, but we're going to get some here. So, why do we worry about studying genetics and blood bank? Family studies are going to help us to predict hemolytic disease of the fetus and the newborn. HDFN, you're going to see me abbreviate that as HDFN, HDN. We're talking about hemolytic disease of the newborn, and sometimes it's the fetus and the newborn. It helps explain the dosage effect. We'll talk about dosage a little more when we get to RH, but it helps explain that. And helps explain any other unusual blood typing results. There's a lot of weird stuff that happens in the lab. You know, everybody thinks, oh, it's just so easy. How many here have ever used a slide to find their blood type in high school or something like that? Oh, a lot of you guys haven't. Okay. We're actually going to some of the high schools and doing some of this because we're trying to like promote our field, right? And so a lot of people think, oh, it's really easy. You just mix this and it'll tell you the result. Well, lots of things go wrong. It doesn't always work well. So it helps us explain when things just don't match up sometimes what's going on. So genetics is defined as the study of inheritance. So when we have a mom and a dad, they each have two sets of all the same chromosomes and one of each goes to the kid, right? I want to clarify right now, when I say mom and dad, I mean male genetic material and female genetic material. Okay, there's a lot of ways for people to become moms and dads. So I'm talking about an egg and a sperm. Okay, I'm going to just shorten it by talking about mom and dad while I'm talking. But just to clarify it, it doesn't always mean just mom and dad. Blood group antigens are inherited traits. So they're not just willy-nilly random. They're dependent on what you get from your parents. Traits or characteristics are inherited from a mom and a dad. A gene is an area that determines an inherited trait. So it's part of a chromosome. It's a piece of a chromosome. It's the part, that gene is for this trait. Each gene is for this trait. The chromosome is the whole thing, and it carries all the genes. All chromosomes carry all your genes. And then the locus is the spot on the chromosome that's going to carry that gene for that particular trait. So genes are composed of DNA, deoxyribonucleic acid, present in the nucleus of all cells. That means, oh, you probably haven't even learned yet. Red cells don't have a nucleus. So red cells don't have the genes. However, they have a nucleus in their bone marrow when they're being formed, and those genes are developed from the DNA. The genes that will tell us what type of antigens are on the outside of the cell, we talked a little bit about that last week. Those antigens are from genes, but the nucleus, they don't actually have, the red cells themselves do not actually have a nucleus, so therefore they do not have chromosomes in them. 4 combinations of bases, adenine, guanine, thymine, and cytosine, and they pair up. Specific combinations determine the different blood groups. Specific combinations and pairings are going to determine, that's the genes, those are going to determine the traits. Humans have approximately 20,000 genes. Actually, I think that's mistaken. If I'm not mistaken, there's way more than that. No, there's a lot. We have a lot of genes because everything in our body is encoded for by genes. We can get abnormal genes. How does that happen? Mutations. We can get a major or a minor change in a gene. Happens all the time. Our genes are always mutating. Minor change, probably not even going to be detectable in the blood bank lab and in the world in general. You know, they just happen. So we get these minor changes. A major change could be more of a problem, and especially in the blood bank lab. It could be more of a problem within the blood bank lab and cause issues with our typing of patients. A deletion, that's where an important part of the gene is just gone. That could result in abnormal typing, abnormal functions of the red cells, things like that. A positional effect is where a gene, if it's sitting next to another gene, is either enhanced or reduced with its expression. So when we get to, there's a couple down the road we get to RH, for example, if you have big D and big C next to it, it makes them weaker versus stronger. So there's things like that we're going to talk about, where if they're next to each other, if the two genes happen to both be there, they're going to actually show up differently. So that's going to be your positional effect. Chromosomes, individual chromosomes carry all the genes, right? Chromosomes, everybody has 2 sets of all the chromosomes, and when we make the gametes, we'll talk about meiosis in a minute, you end up getting half, or the other half, and it's random which half go where. So you get half from mom, half from dad, and that's your new genome. Chromosome 9 is ABO, and chromosome 1 is your RH. We'll explain why that's going to be important later. I'll bring that back up later. But we do know that's where those 2 things happen. The locus is a spot on the chromosome where that gene is going to lie. So the locus is where on the chromosome. Linkage, that's going to be the spot on the chromosome where they're super close to each other, so they tend to be inherited together. Because when we go through meiosis and mitosis, you're going to see how they get divided out. So if they're really, really close, they tend to get divided together. So humans have 46 or 23 sets of chromosomes. 1 set of 23 from mom, 1 set from dad. So this is one person's, like this one might go to the baby, this one might go to the baby, this, one of each of those. And it's pretty random, depending on how they line up and how they split during that process. Every set is numbered except for this X and Y at the end. As I said, the gametes, so the egg and the sperm, each contain only 1 set of chromosomes. We're going to talk about mitosis versus meiosis. Mitosis, 2. Okay? You end up with 2 identical cells at the end. So what happens is the A and B represent chromosomes from the nucleus of the parent. The chromosomes are duplicated. So you got a chromosome from mom and a chromosome from dad. They pair up, they get duplicated, split down, and you have 1 cell and another cell that are identical to the first cell. Mitosis is 2 identical cells at the end. Meiosis has an E, N. Okay? This is going to be where the gametes get formed. We follow it up, the male gametes. A, M, F. So A, this is the person A, chromosome A, from the, I gotta read it, I'm closer, I always get this confused. The mother and the father, so these are the males, these are the female cells, male cells. So chromosome A, on the male cell, this is the one they got from the father, this is the one they got from their mother. First meiotic division, they end up pairing up and dividing off, so you have the same thing, you have 2 of each of them. Then they divide out again, so you end up with 4 cells down here. A cell, sorry, I get confused here. The mother and the father, the mother and the mother, the mother and the father, the mother and the mother. And then over here, we have the cells, the father and father, father and mother, father, father, father and mother. And the sperm, they all hang out. Eggs, we end up getting 1 at a time shot out. It's actually not true. Oftentimes there are more than 1. Only 1 usually get fertilized at a time. So pretty much you end up with some combination down here of half of what's up here. So you can see up here we have some of the moms and dads. Down here they get split up. Some of moms and some of dads. Take some time to read through that. You don't have to understand a lot about it, just know. Mitosis, you end up with 2 identical cells. Meiosis is where you get your gametes, E for egg. Okay? So when it comes to blood groups, we have over 300 blood group antigens that have been identified. You can thank me now, I only make you learn about 15 or so of them. Okay? When you're out there, if you're working like in a research reference lab, you're going to see a lot more of them because the crazy stuff that we can't figure out in the regular labs goes to the reference labs. Most of the ones we use in the reference lab are about 15 of these groups. Blood group antigens are sorted into systems, and the systems are determined by the chromosomes, where the genes lie, the locus of the chromosomes, so where they are, and the characteristics produced, the characteristics of the antigen. So that's how we kind of determine the groups. Something I do want to mention is a lot of these groups were determined like in the early 1900s, and now that we're seeing genetics the way we are, we're actually realizing some of those aren't quite what we thought they were. The same thing's happening in micro, where we name these organisms based on appearance and chemical characteristics, and now we're looking at genetics like, ooh, these are actually in the same family. So things are changing constantly. Try to keep up as possible. You just do the best you can. So we're talking about blood group genetics. We have alleles, alternate choices in the genes on the chromosome. So for example, we all know about A and B, right? If we have A and B alleles, I need to get my drink right top. So for example, chromosome 9, in this person, they got an A from their mom, an A from their dad, and that's what's going to show. This person got a B from mom and an O from dad, and so therefore they're going to show, they're going to have a B and an A, or sorry, an O allele. So these are our alleles. We also have E's and E's, the VAs, B's, B's. We'll talk about those other groups later. Now, homozygous, we're talking about homozygous alleles. We are talking about alleles that are the same on both chromosomes. This is a homozygous A. Homo means same. Hetero means different. So when I talk about a heterozygous alleles, they're different on each chromosome. So this is a heterozygous allele. They got two different chromosomes from the parents. We'll talk about phenotype and genotype next. Phenotype, what you see. Okay? Phenotype, what you see. So for example, when I type you, I can put together things in the tube, look at them, and I can tell you that you are type A. That is your phenotype. I cannot tell you if you're AA, or for example, like this guy, their phenotype is going to be a B, but I don't know if they're a BB or a BO. So phenotype, what I see. Yes?
So would phenotype be like the trait that's presented, and then genotype is the actual genetic
Yes.
material that came to present that phenotype?
Exactly. So genotype, this one is an AA, this one's a BO. So phenotype in this situation gets one letter, unless it's an AB, because they both show up, that's all the different, but for the most part, phenotype, that's a phenotype A. It's a genotype AA. This is a phenotype B because the O is silent. It's actually an amorph. So therefore, this is phenotype B, genotype BO. Like I said, we cannot always tell what their genotype is by what they look like. Dominance versus recessive. Dominant, only one trait is expressed. For example, that BO. B is dominant. That's what we see. That's the dominant trait. Recessive, the trait is not expressed unless it's present on both. Now, with B and O, it's a little bit harder because O is actually not a recessive trait. It's an amorph, meaning it's not there. It's a lack of something that's there. But think of it as recessive. You have to have both of them not be there to know that they're not there.
So O is more like what happens when stuff is missing?
We'll get into it in the next lecture. I can explain it better.
So you know, like if you have a type O, that you know the genotype. That's like the one and only exception.
AB is the other one.
Oh, AB.
If you have AB also.
Because obviously both have to be present.
Yep, exactly. But you're right. If you have an O, you know your genotype is an OO.
Okay.
And AB is the same way. Even though they're
Because
both
it's
dominant,
Because it's
they're
dominant.
both going to show, right?
Yeah.
So those are the two that we readily know. If it's an A or a B, you don't know if your genotype is an AA or an AO, or a BB or a BO. So when I ask you, write the genotype, I expect you to write both of them, unless you can tell. One of the homeworks for this week, you can actually look at the parents and know what the genotype is going to be. Because
Because the genotypes are given.
yes, because we know that we can look, we kind of build the tree backwards and figure it out.
Do we use like those Punnett
Yes,
squares to
we'll get into
determine
those
probability?
coming up.
Okay.
Yep, we gotta play with Punnett squares. I was just doing Punnett squares with my other class the other day.
So I felt like that I learned from like previous like Punnett square things, whatever, that like dominant and recessive genes are like the dominant is more likely to be expressed, but it like the recessive could be expressed.
In blood bank,
Okay.
no, the dominant will be expressed. So see, that's the problem with bodies, is bodies aren't straightforward, right? I think we all can agree with that. And so there will always be those like this, but that, and you will always have things that show up. Just like I always laugh, reference ranges. Reference ranges are great, unless you don't fall in them. And then your normal is wherever it falls, right? So I laugh, my SED rate, for example. My SED rate, normal, 35 to 40 on a regular basis. That's my normal. It's abnormal per looking at it, but my doctor and I go, yeah, that's my normal, you know? So same idea. Things will go wrong along the way, but for the most part, when we look at dominant, we're looking at ones that are going to be showing in with regards to blood bank. Co-dominant, this is your AB that we were just talking about. Co-dominant. They're both dominant, they're both going to show. So if I phenotype this, they're going to be phenotype AB. So therefore, I know the genotype is AB. So Punnett squares, here we go. Told you they were coming up. In this situation, I have a mom that's an A and an O, and a father that's an A and an O. So what can they pass to their offspring? Father gives the A, mom gives the A, they're AA, type A. Father gives the O, mom gives the A, they're AO, type A. Mother gives the O, father gives the A, they're AO, type A. Father gives the O, mother gives the O, now they're type O, because they got an O and an O. So, I always find this funny, because people always get, why can't be an O? My parents are both A's. But here, this is how it actually happens a lot. Thankfully now people can look it up and realize, but you know, back before we had the internet, which was a thing for some of you guys, there was a time before the internet, people would panic and be like, oh my parents didn't, whatever, I was adopted, whatever.
So that means they have to have a O, they can't have a, like both
Exactly.
parents have to have a O.
Exactly. So that's how we figure this out. If I know that my mom and dad were mom and dad were both A, and one of the kids ends up an O, I know that they both had to be AO.
Because each have to give a O gene.
Exactly.
Each of them have to.
This guy, AA versus an AO, we don't know what mom and dad were. We can't tell. So this is where some of the family studies and the genetics comes in handy. Not necessarily for AO, because for the most part, we really don't much care. But when it comes to other diseases, other antigens, other problems, this is where it gets helpful. So RH, now these are very simplistic ones. Yes?
Just going back, so A wins over O, but B wins over, or
B is co-dominant with A.
what about with O?
With O, B wins over O. Everything wins over O. O is like not there. When I get to the next PowerPoints, I'll show you kind of like what that looks like based on the sugars on the outside. So I think that'll help that make sense. But A and B will show over O. O is a non-expression. But A and B both show.
So it's kind of like a zero in computers, it just means off.
Yes.
Just to confirm, if you were a BO patient, you would present as B, but you have the O gene.
Exactly. So you have the opportunity to give an O to your child, and you have an O from mom, but we don't know that unless we go back and look. And like, if you have an O that's a mom, and then she has an O and an O, if she has an O, we know she has an O and an O, so she has to give you an O. So if you're a type A, but you know mom was O, you know you're an AO.
Oh, I was just gonna say, I kind of know what my genotype is, because I know that I'm a B positive and I know that I'm a BO because my brother, I have a twin brother that is O negative. So
Oh,
that means my mom had to have that O gene.
yep, exactly.
Mhm.
So, yep, that's always fun. Twin studies are fun. We learn a lot from twin studies. Okay, I talked about that one. Okay, now we're to this one, Rh positive. So if mom and dad both have a positive and a negative, same thing. You can have a positive for mom, positive for dad, it's Rh positive. Mom is, the mom gives a negative, the father gives a positive, it's positive. Father gives a negative, mom gives a positive, it's positive. But they can both give negatives and be a negative. When we get to Rh, we'll talk more about this. Rh also is either there or not there. So the Rh negative is just not there, Rh positive means it is there. Yes.
So it is similar to the O in which if there was a positive, then it is positive, but only when there's two negatives is it negative.
Yes.